Hey guys, Dr. McDonald again. So we're going to start on chapter 2 for the lecture, which is basically this is a microscope chapter. That's what it is. So let's keep going and see what we see next. The relative sizes of some of these microbes, and it depends. There are viruses which are larger than some bacteria. There are some bacteria you can see with your naked eye. As a rule of thumb, bacteria are a lot larger than viruses. Viruses are extremely small, and as a rule of thumb, we can't see these guys under a light microscope. Now, some of them we can. There's that zero point 0.2 micron micrometer. Some of these guys are. Some of them are too small. All right, let's keep going. Uh, Microscopes are basically magnifying lenses that are placed in series like this. You have one in an objective and one in the eyepiece, and there's the slide. Light comes up, comes through the first, to the second, and then to your eye. And again, this one, the ocular lens is always going to be at 10 times the magnification. This guy can be anywhere from 4 to 100. There are 4 lenses that do that. If it's the 4x, then the total magnification would be 40 because it's 4 times 10. This is an important concept, the limit. The limit for the light microscopes is the resolving power, or how resolving is. I've already said this in the Micro 1 lab, but that's the ability to tell something is 2 objects and not just one. And again, if you were traveling down a road and you see a forest coming up maybe miles ahead, you can't see individual trees. The closer you get, the more it's magnified. Then you can see them as individual trees. That's what resolution is, guys. That limits our eye by itself can only distinguish things that are at least 2/10 of a millimeter apart. This microscope can do 2/10 of a micrometer. Millimeter is 10 to the minus 3. Micrometer is 10 to the minus 6. But anything beyond that is going to be beyond the light microscope. There are other microscopes that take up the slack, though. Magnification, we've kind of talked about that. That's going to be making things look bigger than they really are. Resolution, we've kind of talked about that. That's being able to distinguish things. So if I looked at something and I couldn't tell whether it's 1 or 2, if I can tell they're 2 distinct objects, then I've got a certain sharpness there. Here's something we haven't talked about, contrast. I think I did tell you guys in the first one, problem is bacteria are clear. If you put something clear on a bright white background, you're not going to see anything. Contrast is the ability to see something, like the polar bear in the snowstorm. Hard to see. You need for these guys to have some contrast in their coloration in order to be able to see it. Early microscopes, very interesting, but microscopes can either be simple or compound. Almost all the ones we use in school and all the light microscopes are going to be compound. The simple only has one magnifying lens, and I showed you where we had 2 lenses, one in the ocular and one in the objective. That's going to be the definition of a compound. A magnifying glass is a simple microscope. You can get 3 to 20 times larger using a magnifying or a simple microscope. Leeuwenhoek's had one though that had like 300 magnification, which is pretty decent, thinking he was living back in the 1600s. So the compound we've talked about, and since we use light in order to see these guys, we call it a compound light microscope. And we can go to about 1,000 times, and that's it. We can't go any higher than that. Why can't you go 10,000? Because of resolution. You could make it bigger, but it would just be bigger and blurrier. And if you can't, if it's blurry, then it's not helping you at all. 1,000 times better than your eye by itself, which isn't bad. That enables us to see bacteria. Components, we did this too. Stage is where you put the slide. There's clips there that hold it in there. These are the focus knobs, and what they do is move the stage up and down. That's how you focus. You've got a coarse focus, which is this outer ring, and this moves the stage in large increments. Then you got this fine focus, which moves it very tiny increments. There's the iris diaphragm right there. We oftentimes move that little switch, that arm right there, and it changes the amount of light that reaches here. These are the objective lenses here. There's usually 4 of them going from 4x, 10x, 40x to 100x. This round piece here is the nose piece. It actually rotates and lets you move a different ocular in there. There's the light source. We didn't talk about this in the lab because we didn't have a picture. These knobs are what moves the stage side to side and front to back. So this is the vertical controls, and these are the horizontal controls, if that makes sense to you guys. Oil immersion, we talked about that in the lab, and probably not, hopefully not a surprise to you. The higher you go up in magnification, the longer the objective lenses are. Okay, but the problem is that because of refraction, everybody's seen a pencil, you put it in water and it looks like the pencil's broken. We know it's not true. It's the light waves are bent when you go from one medium, air, to another medium, water. Well, this is what's happening here. It happens all the time, but it only becomes significant when you go up to the highest power. When you're leaving the slide and then it goes into the air before it goes in. So there's the objective lens, there's the slide. You have an air gap in here. It causes a refraction, which means you get a bad image. We put a single drop of oil, and that oil is going to bridge this gap totally, and that's going to keep the light waves moving straight and prevent that refraction. Different types of microscope. Bright field is the one that we do. We look at darkly stained or dyed things on a light background. There's also a dark field microscope where they use a dark background and they have the specimen be light. Phase contrast, all of these are not going to be things you're going to see in a hospital. However, it's a good thing to know them. They may show up on one of your tests. So please read through that and just focus and shoot me any questions. Got some more over here. The electron microscopes, definitely you and I will never see these. These are for research scientists. Electrons, instead of using light waves, they use actually streams of electrons. And again, this is something way over our pay grade. Transmission and scanning. These guys, the scanning is better for the surface But if you want to see good pictures of the interior, then the transmission, you'll see them on a lot of our pictures we see throughout the course. It'll say SEM or TEM, and that's where telling you that's where the photomicrograph came from. And a lot of times they'll artificially add color just to make it visually interesting, I guess. Probe scanning and atomic force. Again, guys, these are high-end microscopes that are not going to have a lot to do with healthcare, but I would spend a little bit of time looking these guys over. Dark field and phase contrast, showing you some of the, whoops, sorry, I was going to try to move that, but I guess it didn't work. Showing you some of the ways that these work. Again, these are not critical for healthcare, but it wouldn't be a bad idea for you to spend a little bit of time looking at them. It's not the highest priority, but it might show up on the final. That's all I'm trying to say. Here's the schematics of the electron microscopes. The electron gun that shoots the electrons, the electromagnets that kind of focus them, keep them going straight. It's a lot. Transmission electron microscopes, you can see a lot of structure there. Scanning electron microscopes, very nicely done. It looks like, well, it's a rod type of bacteria. That looks like DNA. I think it may be DNA. Oh well, the caption doesn't show. How do we prepare a slide? We'll actually talk more about this in the lab class, but just to give you an overview, you generally you grow bacteria on a plate. You take a little loop-like wire loop structure, and you get a little bit of the bacteria, put a drop of water, normally deionized water or sterile water, and then you have this bacteria that's caught in the loop. You put it in there and you stir it up. You let these air dry, which is why you don't want to put too much water. Once it air dries, you light the Bunsen burner here, and you swish it in once, twice, and then thrice. That heat is, you don't want it to stay there. It needs to move pretty rapidly, but that's going to cause these things to stick to the glass, because you don't want these guys, because some of the ways that we color them require us to put liquids on them, and you don't want to wash off your bacteria. Positive stain, negative stain. Gram-positive stains, again, it's abbreviated G+. That's a pathetic G. That's a little bit better G. We'll talk more about those. It has to do with the bacterial cell walls. These guys come out looking kind of purplish. These guys kind of look a little bit pinkish. It tells you something about the bacteria, and very important information. Negative stain, well, they're talking about positive and negative. I'm talking about grams. I'm sorry. We're going to do a lab on this as well. These guys are Bacillus anthracis. These are the guys that give you anthrax. They have got crystal violet, which is a basic positive stain. The positive stain is going to be attracted to the organism, and it's going to attach to them and give them coloration. This specimen has been stained with one that we won't use, and it's a positive acidic stain. Now, these Bacillus megaterium here, notice they look white, and the background looks red. So on a negative stain, you don't stain the organism, you stain the glass around it. So the positive stains are attracted to the organism. The negative stain will be attracted to the glass. Here's our gram stain, purple, blue, blah, blah, blah. This actually is going through the process. I'll go through it for you guys. So the first thing we do is after we heat fix the bacteria, we put a few drops of crystal violet on there. We let it sit on there, I believe, about 60 seconds. Then we rinse it off. Then we put iodine on there. It's going to help the crystal violet to stick to the stain, to the cells. And notice the gram-positive and gram-negative are both purple at this point. Now, this is I think that stays on 60 seconds too. And then the alcohol is only a 20 second. You put it on there and then you rinse it off after 20 seconds. Gram-positive, it's not going to wash off because it's already been absorbed. The gram-negative is kind of superficial. It's going to be washed off. And then lastly, you're going to take another stain called safranin. You're going to stain these specimens with it, but for the gram-positive, they've already been stained with the crystal violet. They will not take it. This guy who had the crystal violet washed out by the alcohol will take it up and will get a pinkish color. If you look over there, you can see the dark purple. Those are spherical or cossi, and then the pink rods or bacillus. That is gram staining. It's kind of an art form, but I think anybody can really do it. It just takes a lot of care and a lot of precision, but anyone can do this kind of stuff. Differential stain and the acid fast, that's how we stain tuberculosis. So there's TB cells, and this Zeil-Nielsen staining has put the TB cells as red and the background is blue. Acid fast, they're just going through acid fast or those that aren't acid fast. This stain is going to make these guys reddish purple. Alcohol is going to wash it out of everything except for the acid fast stain. That's why we call it acid fast, because when you put the alcohol on it, it does not wash away. And then you use a green stain, and these guys, just like we did in the gram stain, since they had theirs washed out by the alcohol, they're going to take it up, but this guy isn't because he's already accepted that stain. Special structure stains, capsule stains. This is one situation where, if you look here, we've stained the actual bacterial cells right there. And then we also stain the background. And if you look, you can see there's the cell, there's the background, and you see this cleared space around it. That's the capsule. So you got a bacteria that has a capsule around it. Helps to protect them and makes them a little bit more difficult for us to repel. Here's one with India ink for the fungi, and you can see that what happens is this stains the background, and it doesn't penetrate into the cells, so they stick out. Okay. Another stain is endospore stain and flagella stain. You guys, these are things you need to know for that lab, especially for the unknown. Let's do the flagella first. It's fairly simple. Flagella are those little things that the bacteria has that helps them move. They can have them all over their body. They can have them just on one end. There's a lot of variety on that. So here's what we do. We call this a stain, and I guess it is, but really what happens is it just kind of coats it. And by coating it, makes it large enough for us to see with our eyes. As a matter of fact, in the actual lab practice, a lot of times it looks like this. I think it's just because, well, none of us are professionals, but the only thing this is going to tell you is yes, this has a flagella, or no, it doesn't. If you put flagella stain there and it does not adhere, then that means that's not a flagellated cell. Endospores, we'll talk more about this. Endospores, there's the regular bacterial cell. It's staining the the pinkish color with safranin. We use a special stain, and we have to use this, actually put it over a boiling uh, beaker of water and use the steam to drive in the green stain into the endospore. Endospore is a cross between a tank and a lifeboat. We'll talk more about it. But what the point is, that what you're doing is looking for these are all steps to help you identify bacteria. Is it gram positive? Is it gram negative? Does it have spores? Does it have flagella? These are the ways we eliminate and zero in on who we're dealing with. And if I hadn't told you guys yet, I think I did in the number one, we need to know this. Not all bacteria react to the same antibiotics. Some of them are resistant. I need to know which one. Yeah, a person comes into the hospital, they're sick. I'm going to start giving them antibiotics. I'm going to give them general antibiotics and basically what I'm doing is crossing my fingers and saying, I sure hope this is the one. But I'm also going to get blood cultures, urine cultures. I'm going to grow whatever's growing in there and see if I can identify it. Because if I can identify it, then I can, at the same time, they're going to tell me which drugs are most effective against it. So then I'll switch to the one that's really going to give the knockout blow. All right, guys, let's see. You guys can do this review on your own. It's pretty good. Go read through this and see what you can see. All right, guys, that's it for chapter 2. That's everything for the first week, and I'll get started on some of the others.